ended up being a session by my very long-term colleague Reinhard and myself, and it was sort of open maybe your view on other aspects of the cell and the components may be important for cell migration, and admittedly, both of us know very little about it, but that's why we're here. Well, Reinhard, you forward.
Okay. Welcome everybody, and the topic of my talk is the dynamic cytoskeleton, which is a quite broad topic. So I made some subtitles talking about the different jobs of actin. Of course, we have heard a lot of actin already. Microtubules. I'd like to make a short introduction into microtubules. And the main topic of my talk will be intermediate filaments, the third part or the third group of filamentous cytoskeletal elements. And we'll focus at the end of the talk on the role of the intermediate filaments in cell migration. Just an introduction to the cytoskeleton. The cytoskeleton is a very prominent part of the cell. It keeps basically the cell alive by maintaining cell homeostasis. It's one of the duties is transport or cell division. So lots of basic parts of in the cell life are arranged by the cytoskeleton. We'll see more examples later. And the second part of the cytoskeleton we're talking here about is, of course, cell migration. So cell migration is, in either way, the movement of an entire cell, of a single cell, or, and Rudolf will tell you more about it, the movement or the migration of groups of cells, which is also very important and maybe very prominent in a living organism like we are. Cell migration is, we have heard about it used in development, especially, or the main example is brain development, where every single neuron has to get its perfect place in the brain so that we can stand here and talk and tell intelligent things. But it's, of course, also true for other organs. They have to be, cells have to be arranged in a specific manner and they have to move to their places. Immunity is very important. Cells move. You have seen this movie before several times of the leukocyte cells moving through the body and tracking for problems. Wound healing, this repair mechanism, especially important for epithelial cells, which we will specialize on later on. And the problem with cell migration, this is cancer. Cancer cells tend to migrate out of the primary tumor, building metastasis, and understanding metastasis, cell migration metastasis is a really hot topic and still very poor understood. It's not even clear if it's a single cell which is migrating out of a tumor, or if it's a group of cells migrating out of the tumor, and perhaps it will be both. There's a question.
Have you got your microphone on? Because it's quite hard to hear you back there.
Okay. Is it better now? No, it's not better now.
Okay, there you go.
Okay, here we go. Okay, I will not repeat it. So, I made this comparison between a driving car and a migrating cell, just to show what are the duties of the different aspects of the cytoskeleton. So we have the actin filaments. We've heard about the actin filaments. These are the motors. If you think of a car, the microtubules are something like the wiring in a car, so they send signals from the driver to the motor to the lights and so on, and maybe even back if you have a modern car. Of course, the intermediate filaments, they form the chassis, so this is a scaffolding for the whole car, holding everything together. That's why they're so important, of course. There's a fourth group of cytoskeletal filaments. It's septins. The septins are kind of gaskets which seal the fuel or the water against the rest of the car. I will have one short slide just to introduce the septins, so you keep them in mind that they're still there. And the car, of course, has contact to the surface. These are the tires, and in our case, are the cell matrix contacts. The car wants to move the driver, or the driver steers the car.
Nucleus is not the driver.
The nucleus is not the driver. We discussed it with Rudolf. Who's the driver?
Small G proteins might be driver.
Yeah, but where do the small G proteins come from?
I propose we don't discuss that into depth, because otherwise we'll spend the hour, I'm sure.
Yeah, but this might also be a kind of model.
It certainly is a model.
It's certainly a model, and it has its limitations. So just repeating the actin story with adding some aspects we haven't heard about it the last days. So actin, in one way, is a rigid structure, or it can be organized as a forming rigid structures. These are the microvilli, or the cell cortex, or stress fibers. It should be clear that these rigid structures means that they still can be reorganized and are not as rigid as a concrete, something made from concrete, because it's a living system. But it's relatively rigid, and there's strong interconnection between the filaments. The other mode is kind of motoring the cell. This can be done either by motors, which are the myosins, or by polymerizations. And different kind of we have lamellipoda, invertipoda, podosome, and so on. So this is a actin classification. We already heard about the actin polymerization. We have the soluble G-actin, which is a monomer, binds to ATP, forms dimers. These dimers can polymerize into these helical strands, and these are the actin filaments we're talking about. The actin then is released, and in the next scheme, I'm still repeating this scheme, everyone has seen it here now. So here we have the actin monomers. When ATP binds, dimers are formed, and dimers can form filaments in two ways. One way, in the Filipodia, which were introduced by Sasha yesterday, with the formins at the tip connected to the membranes, and they're forming this stable, not really stable Filipodia, but this higher connected bundles of actin filaments. And the other way is via the R2-3 complex, where network is formed, which pushes somehow the cell forward. We should keep in mind that it's not only a pushing forward of the actin lamella, or of this resulting actin sheet. That depends on the adhesion of the cell to the surface. When there's no propagation of the cell, this whole actin polymerization can take place, and the whole actin sheet can flow backwards, which is known as retrograde flow. And if there are new connections formed, then this whole lamellipodium may move forward. If we look in detail here, we have this Filipodium introduced by Bernd yesterday, and behind with its bundles of actin, about 1 millimeter space behind this so-called leading edge, we have the so-called lamellipodium, where we have this network of up to 3 branched actin filaments. And it has become clear that this lamellipodia is a relatively small, narrow part of this of a moving cell, and that in the region behind it, here is 10 to 15 micrometer, but that depends a lot of the different cell types. There's a region which is called the lamellum, and within this lamellum the turnover of the actin filaments is quite different from the turnover of the actin filaments in the lamellipodium. Notably, the actin filaments are much more stable, but there is a clear border between the lamellipodium and the lamellum with more stable actin fibers. We're not talking about stress fibers here yet. And this whole complex is attached to the focal adhesion family. In the very tip, there are these nascent, short-living adhesions, followed by focal complexes, which are more or less located at the border between lamellipodium and lamellum. And finally, the major focal adhesions, which are the sticking points of the cell to the surface. This lamellipodium, I introduced the lamellipodiums as they had contact to the surface via the focal adhesions, but in this, or the focal contacts, focal adhesions are far back. If in case there is no adhesion and there is still formation of new actin filaments, this leads to the formation of so-called ruffles. Here we see and ruffles means that the polymerizing actin somehow does not move in the flat direction of the surface, but makes this kind of curvature to the outside of the cell, and this can propagate back over the cell, even can propagate over the lamellum. There are also dorsal ruffles, and in this movie here we nicely see ruffles. In this case, now the movie is starting, there are no ruffles, and after a certain time, we see a chymogram here after 5 minutes. Sorry for not having the time here, but after we see here in the chymogram the first 5 minutes of this movie, and we see that the edge is straight, and after activating RUC, this small GTPases, we had a lot of this morning, the ruffling starts, and this is clearly seen here in the chymogram. And on the normal microscopical image here, you see no rufflings, and now these black lines here, they correspond to these ruffles seen here in this cross section. So they appear somewhere at the border, move a little bit back, and then they disappear.
What are the vesicles that you see moving retrograde from the edge of the...
I have no idea. When you have a living cell and you have a wide field image, you see lots of structures within a cell, and it's nearly impossible to judge what it is.
Towards the bottom there.
Oh, this is
This one?
macropinocytotic.
These ones?
Asking you.
Yeah, maybe these ones. Yeah. Yeah, here. And these other vesicles are hard to decide. Sometimes you see very clear mitochondria in the lamellum, but in this case, not. Yeah. So in the front we have the lamellipodium, lamellum ruffles. Then we have the dorsal arcs, introduced by Sasha as transversal arcs, which are these band structures here. And finally, I will not tell more about them. And then we have the stress fibers reaching from the lamellum to the back of the cell. The ventral stress fibers reach focal adhesions in the back of the cell, while the dorsal stress fibers end up without connection, with any known connection to other structure. We'll see it later. This is a movie showing these details. You see at the very border, at the top, you see the lamellum. So for some reason it's not repeating. It's a very so this is an actin staining, and you see that the border of the lamellum is a very narrow band very close, representing the edge of the cell. This is, and going more inside, there is a darker region here. This corresponds to the lamellum. Sorry for this, but PowerPoint is not improving this program. It's getting more worse over time. You agree? Here you see the transversal fibers moving backwards, and
I operate this for you. Wait a second.
it's not a question of operating. You can take the mouse. So you see this. This is a lamellum, the lamellipodium, the very front end, not very short in this case. The fibroblast behind the lamellum. There's the dark region. There is still inward flow. You see it very nicely here on this transversal arc, so dorsal arcs. And here you have the this will be ventral arcs in this case because they're not reaching over the whole cell. You're fine?
So this is keratocyte.
No, it's not keratocyte. It's a movie from Lapalainen.
It's Lapalainen and it's U2 osteosarcoma.
Yeah. Yeah. Okay.
So you don't care what's happening at the back. It's quite interesting. I mean, there's quite a lot of time.
I would interpret it that this cell is not moving. It's just a stationary cell, and it has more or less two lamellipodia, but it's not approaching a certain direction. The other structure we have to think about, or the second filamentous system in cells, are the microtubules. Microtubules have a special organization. Basically, they arise from a microtubule organizing center, which is close to the nucleus, and on the outside worded there is a plus end, and the minus end is connected to the microtubule organization center. And as these microtubules are built up of two proteins of tubulin, this structure is a polar structure. So this is why we have a plus end and a minus end. And since and this gives the cell a direction. It gives a direction from inside to outside. So if you move along a microtubule, you will always reach the center of the cell. If you move outside, you will reach the outside of the cell. So the microtubules are somehow the architecture network for the architecture framework for the cell. But there are also stable forms of, this is normally somehow stable in cells. There are other stable components of microtubules. These are the cilia and the flagella. We heard about the flagella this morning. These are these long structures, moving structures on the volvox. Individual cells, cilia are somehow the same, but they're just shorter. Extremely important is a microtubule for cell division, because the microtubules are the structures that move the chromosomes into the daughter cells. So before the cells divide, this microtubule organizing center divides into two parts called kinetochores. Then they connect to the chromosomes. Chromosomes are arranged, and the microtubules push the chromosomes into the daughter cells. There's also a pushing and a pulling event in this separation of the chromosomes, and this is an astonishingly
fast process goes within minutes, so very fast. And this network of microtubules, which is defined within inside of the cell and outside of the cell, is used by motor proteins which transport different, which are responsible for the positioning of many things within a cell. So kinesins transport material to the outside and dyneins to the inside. Of course, there are exceptions, like always. But for example, intermediate filaments, here in blue, can be transported in either direction. Actin can be transported along these microtubules. Microtubules can also be used for the attachment of actin, as shown here, also by dyneins, but they are not moving in this case. Also, the positioning of microtubules or other organelles is maintained by these motor proteins, and vesicle transport is a very prominent example also for the transport performed along microtubules. At microtubule plus end, there are lots of different proteins which also are maybe catalyzators for certain processes. So these are kind of reaction centers. We will see that when they reach focal adhesions, that they can influence the turnover of focal adhesions and therefore modulate the activity of focal adhesion, so the adhesion of the cell. Here's another cartoon showing all three filamentous systems. The coloring is not so nice, but I will lead you through it. So the intermediate filaments, so this is basically a lamella of the cell. We have here the lamellipodium with a ruffle here, which is empty in this case. The microtubules are these grayish structures here, leading into the tip of the lamellipodium. This no color filaments, and also here, these are the actin filaments. This is the actin network. And here are actin cables, the stress fibers, and intermediate filaments are reaching into the lamella, but not reaching into the lamellipodia. And different duties are performed by microtubules in this situation. There's cross-linking between the different cytoskeleton elements, so microtubules are connected to each other. Microtubules are connected to IFs, not directly. IF is abbreviation for intermediate filaments. They are not directly, there's no direct interaction between microtubules and intermediate filaments, but there's a cross-linker, it's called plectin. Or there's also interaction between microtubules and actin, for example, via alpha-actinin. The traffic, already talked about, vesicles are transported heavily along microtubules, and other examples are intermediate filaments, or for example also RNA may be transported along the microtubules. And here in this zoom in, not shown here but shown here, we see the tip complexes. The tip complexes may contain guanosine exchange factors. This may activate RAC, and this may then also perform induce actin polymerization. So many different kinds of actions performed by microtubules, and every community tends to blend out some other intermediate cytoskeletal filaments. Microtubules should not be forgotten somehow. But they are extremely nice. In this cell, in fact, when you look on the microscope, there are sometimes they are nice, sometimes there are too many microtubules in a cell that you do not see any individual microtubules. So if you have the right cell and the right labeling, in this case it's rhodamine labeling, so the dye was injected into the cell. You see very nicely how the microtubules grow towards the cell center. So this is a lamellipodium, just the border of the cell here. And you can clearly see how here single microtubules grow, stick, or touch the surface of the cell, and then disintegrate again. So microtubules are under certain conditions can be very unstable, so they grow and they tend to depolymerize quite fast. So this is called dynamic instability, and this is what we see here. And this is kind of probing of the cell surface here, and in certain cases then they may start some actions. Definitely.
It seems that we also see some global retrograde flow, a bit like for actin in this case. Is it a common feature of?
Yeah.
Okay, so it's general.
If you look for a cell which is somehow more, which has some lamellipodia or so, and you look for different structures in a cell, you see mainly inward movement of many things. Also for keratins, as you will see later on. So this general actin backward flow also of the arcs, many things are connected to this flow and follow this flow.
Okay, so this is dependent on actin retrograde flow, the one we see here.
It's a question what we define as actin retrograde flow. So normally, when people say actin retrograde flow, they respond, or they think about the lamellipodium. So there are different modes of actin backward flow. This is the lamellipodium, where the R2-3 filament is moved backwards. Within the lamellipodium, different mechanisms are observed, and then you have also the backward flow of the transversal arcs. So there are different mechanisms for the actin backward flow, but they all result in a somehow net inward moving of everything, which cannot be true. Otherwise, the cell would be round within half an hour. So there must be also an outward movement, but we will have the same problem later on with the intermediate filaments.
These are bundles of microtubules?
No, these are individual. These are individual microtubules. That's right, microtubules. So the diameter of a single actin filament is about 8 nanometers. Microtubule is 50 nanometers. So basically, for light microscope, same size. But of course, in a light microscope, you can see things that are smaller than 200 nanometers, which is the resolution limit. You can see it when they're individually. You cannot judge if it's one or two, but if you see many films, you can be sure that it's a single microtubule. When you see actin filaments, normally you see bundles of actin filaments. The same will be true for the intermediate filaments. But under normal conditions, microtubules do not form bundles. They may form bundles in the case of the Volvox. There was kind of cortex of the microtubules, or in the sperm there are bundles of microtubules. But these are individual filaments. These are the septins. Just to remember that they are, or to deduce them, there are at least about 13 genes in humans. They form heteropolymeric complexes which can form filaments. These filaments can even bundle and tend somehow to form rings. They are involved in the establishment or the maintenance of membrane diffusion barriers. Examples are a cilium, a single cilium, especially the primary epithelium, the primary cilium and epithelial cells. At the base of the cilium, there is such a septin ring underneath the membrane, but this maintains two different plasma membrane regimes, inside the cilium and outside the cilium. Same is true, for example, for a sperm. In the flagellum, you have a different lipid composition than in the rest of the sperm. Also, on these bladdings on dendrites of neurons, these septin rings are found, and first they were found in a budding yeast. Yeast cells divide by making these sister blobs, or buds, how they call it in yeast, and this is a kind of ring which is also, or septins are involved in the maintenance of this ring. But let me come to the intermediate filaments, which is a quite large group of proteins. While actin is simply by G-actin, or microtubules only by two different proteins. Within the intermediate filaments, we have more than 70 different proteins in humans. Like actin microtubules, they also build a cellular network, which is somehow different. And the main function of the intermediate filaments are mechanical functions. And most important are these mechanical functions if tissues are formed. When you think of our skin, skin consists of many cells. Cells are somehow connected to each other. But when you stretch your skin, skin is extremely resilient to stress. And this is due to the mechanical functions of this cytoskeletal component of the intermediate filaments. They cannot do this simply by being in a cell. They have to be connected to the neighboring cell. This is done by cell contacts. Rudolf will introduce them here. So this is a main function. This is one of the functions of the intermediate filaments to maintain tissues. What's special about, or what's different to the other filament systems is that we do not have any motor proteins which use the keratin, the intermediate filaments as tracks. The intermediate filaments can be transported, but they are not used as tracks like the myosins or the actin or the adenosines and kinesins on the microtubules. So there are 70 different proteins, which makes it quite complicated. There are, I will tell you something about the classification. We have 6 types of intermediate filaments. We will stick further to the type 1 and type 2, that are the keratins. Keratins are the intermediate filaments of epithelial cells, so cells that are on the surface of our skin, for example, or on the surface of the gut, or forming glands that are derived from surfaces like the liver or the pancreas. I will going to come to the keratins later. Second type, or in this case already number 3, is vimentin. Vimentin is basically the brother of the keratins. So most of other cells, for example, fibroblast endothelial cells, contain vimentin. In the main difference between keratins and vimentin is that vimentin is a single protein, whilst in keratin you have how much is it? 54 different keratins. Next, another important member of this group is desmin, which is found in muscle cells and important to give the muscle cells its structure. Also, for example, in heart muscle cells, not only in skeletal muscles. Then there are other types, for example, GFAP in glial cells. I will not go into tell more about it. Another important group are the neurofilaments. There are 3 different types, light, medium, and heavy chain neurofilaments. They are found in neurons, may be involved in neurodegenerative diseases. some other in this group. And next important, or also very important intermediate filaments are the lamins. Lamins are intermediate filaments. Laminin is a part of the extracellular matrix. Don't mix it up. What's called laminin is outside. Why these intermediate filaments are located in the cytoplasm of cells and form their networks, lamin is located within the nucleus. And it does not form three-dimensional network. It forms a sheet underneath the nuclear envelope, and so it gives some structural rigidity to the nucleus. And it's also important for the positioning of DNA within the cell, so it becomes important in regulation of gene expression just by arranging the DNA within the nucleus. And this is the most ancient protein in this protein family, so the others are derived from the nuclear lamina. Nuclear lamine. Okay. Type 6 are special proteins just found in the islands. The keratins are the biggest group, and that's what we will deal now. There are two types of keratins. Keratins within epithelial cells, within the cytoplasm, also called cytokeratins. And there's a second group of keratins which form our hair or fingernails. So they are extracellular, also they are they are forming extracellular structures. We will stick to, of course, to the epithelial located keratins. And you see here the number. We have 70 keratins from type 1 and 20 from type 2. So these epithelial keratins are divided in two groups. This is important because these groups form heteropolymeric filaments. So in order to make a keratin filament, you need one protein of, or even more, you need a same amount of type 1 keratins and type 2 keratins. Type 1s are the acidic ones, type 2 are the neutral basic ones. The nomenclature is somehow confusing because it's historically grown, so the numbers are somehow mixed up. There's been a revision some years ago. So if you take two keratins, for example, keratin 18 and keratin 8, they will form a filament system, and in this case, these two keratins, they are expressed, or they are found in simple epithelium, for example, in the liver or in the gut. Other keratin pairs are found in the epidermis, so surface of the skin. For example, in the basal layer of the skin, the pairs K5, K14, or in the upper layers, K1 and K10. So this is a quite complicated differentiated expression pattern. We do not really know what's the reason for it. It's useful in cancer surgery because when there is a cancer metastasis, you can judge from the composition of the keratins within the cancer cell from which epithelium it originally originated. Okay, this is what a keratin molecule looks like. Basically, it has an alpha helix, which is called the rod, which has a length of about 45 nanometers. And basically, we have a coil 1 and a coil 2. Coil 1, they are separated by a linker element, which is seen here. And coil 1 also has a linker, which is a little bit hidden here. On the N-terminal, there is a head. This is not, while this rod domain has been crystallized and the structure is well known, the structure of this head domain and also of the tail domain are not really known because there are somehow they are folding unpredictably.
Where are the charge groups?
What do you mean with charge groups?
Somewhere acidic and somewhere base.
They are distributed all over.
Uncharged more.
They're distributed all over the proteins. So
Is the net charge? Yeah? Net charge.
They lose count.
It's net charge, yeah. It's actually on the surface of the alpha helix as well. So.
So this is a monomer. There are no free monomers within a cell. This is where the partner comes in, the hetero dimer. So in this case we have K5, K14. This form this parallel dimer. And the rods are somehow intertwined over each other. The head and tail region are somehow speculative in this case. So here we have two heads in this case and two tails, so parallel organization. And the next step in polymerization is the formation of a tetramer, so two dimers, but now antiparallel, build a so-called tetramer. And the tetramer, as the tetramer has been formed from two dimers in an antiparallel fashion, we have two tails on the end, and this whole structure is non-polar. And these tetramers are thought to be the soluble and stable building blocks of the keratin filaments within the cytoplasm. They are diffusible within cells, and as I said, these are the building blocks, the monomer, not the monomers, but the units from which the filaments are formed. And there's a step from in between, from the tetramer to the filament. This is a so-called unit length filament. So 8 tetramers will form such an ULF. This ULF has a length of about 60 nanometer. These are observations made in vitro, mainly by the group by Harald Hermann, where they look under electron microscope. They find these ULF structures. And these ULF structures then, if you have several of them, they elongate into a kind of filament. As this filament here looks like a spaghetti and is quite straight, this is not really true. When we look for the flexibility of these filaments, we see that they're in comparison, so these are the intermediate filaments, in comparison to actin and microtubule, the persistence length is quite short. It's just it's below 1 micrometer, whilst actin and microtubule are about 10 or even about 1 millimeter. So this means that intermediate filaments are quite flexible within a cell.
What is the cross-sectional diameter?
It's 10 nanometer.
It's a little more than of actin.
It's a little more. It's a little more than actin. It's a little less than microtubules. This is why they called intermediate filaments.
Yet they are more flexible than actin.
Yeah, they're more flexible there, and they're also more elastic. So microtubules and actin are somehow have the same elasticity, which is close to the other keratins, to the hair, interestingly.
Then bending elasticity or twisting, which kind of elasticity? With respect to which deformation?
This is most likely this is effective modulus that you calculate if you recalculate bending into
Into
the rigid body model, bending into
shear.
an
Shear.
So in comparison to the microtubulous actin, the intermediate filaments are much more weaker and much more elastic. Which is interesting because in the beginning I said these are the mechanical stabilizers of the cell. But they will not break as soon.
Could it be due to links between subunits? Because there seems to be a strong interaction within the subunit, then the subunits.
Yeah, the model I've shown in the animations is very simplistic. It looks like we do not have a strict diameter of 10 nanometer. That the number of subunits is not just 8 or something like that, so that it is variable. And even if you stretch intermediate filament, maybe they will slide along each other.
Some papers show they can be extended up to 3 times the initial length, and because of the sliding inside.
Yeah.
They don't fit around like this.
We made some attempt to measure it. We put a magnetic bead into a cell and electromagnet close to it, so generating a magnetic field, switch the magnetic field on and off every 5 minutes, and look for the displacement of the magnetic bead. Unfortunately, this is not moving here, but you have a diagram here showing that this is I think 1 micrometer, that every time the magnet is switched on, the bead displaces. When it's switched off, it relaxes somehow. This is several events being shown of each other in different cells. We have the wild-type cells, which have normal keratins. We have knockout cells from Thomas Magen, which do not have any keratins. And we have rescue cells where we reintroduce the keratin. And when we switch off for 5 seconds the magnet, there is an elastic and also an plastic compound of the curve. In the case of the green, this is a wild-type cell. It's we reach up to this height. This is somehow showing the displacement regulated by the force. And when we remove the keratins, we see that the cell becomes much more, even much more elastic in this case, even if the keratins are removed out of the cell, which is somehow puzzling. Anyway, even when there are no keratins in the cell, it becomes much easier to remove the magnetic bead out of the cell because the cell becomes weaker, which is due to this plastic compound here. This is something about the charge. It's not the metal charge of the one molecule, but we have intermediate filaments are heavily phosphorylated, especially on head and tail domain, but also on the rod domain, and also other post-translational modifications are present in keratins. So there's glucosylation, there's ubiquitination, acetylation, and so on, making it quite difficult to judge in which state the keratins are and what is regulated by all these processes. As I said, it should be clear that the keratins form a net inside the cell. The net looks like is connected. There are no many free ends at the edge. Normally, there do not appear free ends of the network. And the work of Fabian Herb, he made it by segmenting the filaments. We could generate a digital geometric representation of the network, which means that for a single filament here, which is a filament bundle, we get digital numbers saying what's the length of a branch, how high is the branching, and also the thickness of a filament. And when going for cells, comparing cells, control cells with cells where plectin, which is the linker between different cytoskeletal elements, we see that there is a difference in branch lengths. So the network composition can be changed within one cell, and it's also different in different cells.
Is it an impression that you have mainly three-way junctions there? Or is it true? I mean, in this network, it seems that
Three-way junctions.
Three-way junctions.
Three-way, yeah.
Mostly are three-way.
No, it's nearly almost three-way junctions.
Meaning that you don't have just links between filaments. You have branching.
It's branching, yeah. It's branching filaments.
Yeah.
Interesting. Do you know the structural background for this branching?
No.
And how stable this 3-way junctions?
It depends on the time scale.
But they do decay.
They do decay. They merge each other, or a branch can move. So what we often observe is that two filaments merge to each other. I will show you movies. So this is just an impression of the cell when the keratin filaments are labeled. And going into detail of this whole process, which I will now describe, we will see how in a living cell keratin filaments are formed. This is a cell border. This is a movie, and these are different time points. And what we see that in the edge of the cell, there is a appearance of fluorescence, which means this is a nucleation event of a filament formation. So these three arrows represent the new formation of these nucleation events, and of course the filaments are then they elongate. So it's seen here. It comes from nothing, gets longer. And next step is that they still elongate. These are the three from the movie before. They elongate here, and they merge to each other. So they merge to each other, end to end, always end to end, or nearly always end to end, and then they become part of the underlying network. So first filaments are formed, then the filaments are connected to the existing network. Looking further inside the cell, we see that this whole we again have this retrograde flow. So this is the outside, and this is a newly formed filament, and we see here how they move inside. So there's inward flow, and there's another process. This is bundling. We see here we trace one filament, and we see that this filament is becoming thicker and thicker and thicker. This is we do not really know what's going on there. Are there filaments bundling, or is this single filament just somehow growing? This is still the problem we have with light microscopy.
How far is this from the leading edge of the cell?
A few micrometers.
So this is a lamellum.
This is somewhere in the lamellum. It's between the lamellum and the...
It is probably due to centripetal movement of actin and myosin, yes?
It's simply, yeah. We have not shown it, but the speed is somehow the same.
But at the same time, there is a assembly. So it is
on the outside there's an
because of
assembly.
actomyosin, but assemble kind of during this movement.
Yeah.
And nucleation happens in the lamellipodium.
Close to the lamellipodium. I will show later that it might be close to focal adhesions, but it will not be directly
to the boundary between lamellipodium and lamellum.
Yeah, we have not really looked into this boundary yet. But somewhere in the very close to the lamellipodium, this nucleation appears. And going further inside the cell, here the nucleus will start, or this here, the nucleus, we see that the filaments disintegrate. So following this individual filament, we see that it's after 15 minutes, it's quite a long time, but it's really hard to follow in a movie because it's a 3D structure we are following here. It's not, you cannot follow this in a single plane. So you see this individual filament is becoming shorter and shorter and also thinner. From all these observations, we propose the keratin cycle of assembly and disassembly. So starting again with the nucleation in the very end, in the very outside of the cell, close to the lamellipodium, close to focal adhesions, we have this nucleation of the keratin filaments. We have the elongation, integration into the network, something we call bundling, or so it's also there's an increase of fluorescence, massive increase. So it might not only be bonding, it might also be incorporation of tetramers into the existing network. When we come close to the nucleus, there must be something of maturation because we have a stable cage around the nucleus, and as we observed here, we have a disassembly. So keratin tetramers will go into a solution, and we think that at least to some kind diffusion is the main mechanism of transporting the subunits back to the edge of the cell where nucleation can start again.
At which stage the heterodimers are formed?
They are formed outside this circle before. So just after protein biosynthesis, the tetramers are formed. They're not inside this cycle. Yeah?
So always here you observing the assembly from which building blocks you think?
From tetramers. From tetramers.
Tetramers are.
They may be.
Is the bundling dependent on phosphorylation of the keratin?
This is what we think. We think that phosphorylation is the main trigger for the steps, and actually we're trying to identify several triggers by looking for mutants which have been by pseudo-phosphorylated mutants.
Okay, can you measure the diffusion rates?
We have measured it.
Is it fast diffusion, fast transport, or
it's a normal diffusion of a protein in a cell. So it's quite fast. On small distance, diffusion is fast. But we have not calculated the rate, if the rate of diffusion is sufficient to fuel the formation of the filaments. Something we could do. We made some image analysis. We're not going to the detail. When we're looking for a round cell treated with EGF and map the speed of the inward movement, we can increase the speed of the inward movement, and calculating from the speed and the fluorescence intensity not shown here, we can identify sources, so assembly of keratin filaments in the outside and sinks in the inside. So disassembly of the keratin filament system. By making it by rounding up the cells in the computer, we are able to averaging high numbers of cells and can nicely show how we have can nicely show effects of, for example, EGF, or the time cells are in culture, then the speed is decreasing. We tried to use plectin. If it has an influence on the speed, it doesn't have, unexpectedly didn't have any influence on the speed. So we have here a method to calculate assembly and disassembly within the cell. This comes to Nika's question, where until now we were able to track the formation of keratin filaments. So the green filaments here, this is a focal adhesion, quite a strong enlargement, and we see and could statistically show somehow that the keratin filaments, at least the initiation, takes part in very close vicinity to the focal adhesion. This might be to some mechanical scaffolding or some triggering by phosphorylation or something like that. Keratins are I do not have so much time.
None?
None.
But I think we can, I don't know how you feel about it. If we should.
You have another 15 minutes, sorry. Yeah.
15 minutes.
And Tiga said.
We all agreed on that. We agreed on enlarged.
Okay. Keratins do not only have mechanical functions. This is just a little bit going away from the dynamics. There are lots of functions associated to intermediate filaments and also to keratins which do not have any to do with the mechanics. So, for example, if we have mice which do not have any keratin 19 knockout mouse, and when we add chemotoxic stress, they develop liver fibrosis. So, K19 somehow prevents or rescues liver cells from chemical stress. Also, osmotic stress, keratin seems to be involved, or better in the reduction of osmotic stress. So, keratin is seen as stress protectors, and stresses might be quite unspecific. One idea is the so-called sponge theory. The keratins are highly phosphorylated or have many phosphorylation sites, and within stress responses, phosphorylation is very important, and maybe the keratins just take out a lot of phosphorylation before the important kinases for apoptosis or something like this are affected. Also, the cell cycle may be affected on a certain pathway. There's a connection from the actin to Src. This again is something to do with migration, because Src may activate focal adhesions. I already told something about plectin. This is again something mechanical. This will affect the site architecture, but also has something to do with stress resistance. And also the positioning of the nucleus is maintained somehow by the interaction of the intermediate filaments shown here and the nucleus shown here. Here's the lamine showing, the group I introduced earlier. And there's an interconnection between the intermediate filaments via plectin and via nestrin to the membrane of the nucleus, and therefore somehow the nucleus position is also affected or maybe regulated by the intermediate filament system. There are lots of diseases known which originate from at least mutations in keratins or in intermediate. These are only keratins. From the keratins, the problem is that there are as we have so many different keratins, and which are expressed in different parts of the body, there are many points where diseases can appear. Quite obvious diseases related to the skin, where the integrity of the skin is somehow affected. This may cause somehow looking here like a fish type skin, ichthyosis bullosa, when K2 is affected, or EBS, epidermolysis bullosa simplex, which leads to blistering of the skin, which leads to inflammation, which can be quite severe and can be deadly as long as you do not have any antibiotics, or PC, pachyonychia congenita, which leads to hyperkeratosis. So people have so much keratin on the surface of the skin that it becomes impossible for them to walk. And other keratins which are located within the cornea of the eye also may form kind of cysts. This is an ophthalmologist's image, so that you get visual problems. And besides this, there are also problems appear with inflammatory bowel disease. So these are the keratins which are in the gut or within the liver. They're also candidates for causing diseases. So quite a lot here. When it comes to migration, I showed single cells. The cells I showed you in the cycle, they did not migrate. They stick on the place. They did not migrate. And next step will be, and also in the interim program, to look how this cycle develops when the cells are migrating. Normally, or in most cases, cells are in a tissue, and then they are connected to the basal membrane via hemidesmosomes or by desmosomes to the neighboring cell. In this case, the keratin filament network will be much more stable than in the situation I showed you. But at least in cell culture, when you have a connection to neighboring cells, this cycle still remains. Maybe it's maybe different in living tissue, something we are looking for. And when you have metastasis or wound healing or something like that, then the stable situation will switch again to a migration regime. This is a, will it start? No. This is a mouse epithelial cell with keratins inside. We see here the wide field image with a big lamellipodium. Here's the higher magnification. This is the edge of the lamellipodium, has been somehow registered not to walk out of the image. And you see very clearly at the edge of the cell where these small pieces of keratin appear, move backward much faster than the non-migrating cell. And on the top here, they are integrated into the network. The filament formation starts quite outside, or quite narrow to the edge of the lamellipodium.
Is that intensity gradient in that picture just due to the thickness of the cell, or is there actually a higher density per unit volume of the cell towards the posterior of the cell?
You mean this gradient?
Right, you see it's brighter in back, but that could just be because the cell's thicker in back.
Yeah, it's both.
How does the actual density of keratin change per unit volume, the amount of keratin per unit volume as you go anterior posterior?
Cannot, I have no numbers, but it's increasing a lot. So at the very edge, there's really very small amount of keratins, and the bulk of keratins, they will be in the center. But they're also involved in the turnover. I have another movie showing this cell, this INSEM consortium will deal with. This is the primary human keratinocyte. Here you see a migrating cell. This is this cell type. And we already have one movie with this cell type with keratins. And what you see here, this is the nucleus. This is the front edge, the lamellum of the cell. And you see here clearly new formation of keratin filaments while the cell is migrating to the left. A stable cage around the nucleus, and there is still some nucleation of new filaments at the rear end of the cell. Same cell is shown here again. Without moving, it's the same movie. So clearly you see this inward movement. Then here becomes quite complicated, but there also will be disassembly of filaments. And on the edge, on the rear, we do not really know what's going on here. So bringing all this together, we have a cell which is migrating. At the beginning, at the end, to the left, we have the lamellipodium, we have the lamellum, we have the cell body, we have the rear. We have the microtubules, which are dynamic unstable, probing the cell, giving signals, transporting material to the edge and also to the rear. We have the nascent adherents, which are very short-lived and probing the material. We have the focal complexes, which are short-lived, but maybe by some interaction with actin and myosin become long-lived matrifocal adhesions, which then are responsible for the stable anchoring. And at the rear, we have the rear focal adhesions, which will disassembly somehow by actin-myosin contraction, dephosphorylation. Calpain was mentioned by Bernd, and the whole story will be as something to also with microtubule targeting. This is the actin story. We have the single filaments, the up to 3 filaments at the front, also the filopodia not shown here. We have the dorsal stress fibers, which originate from the focal adhesions and end here somewhere behind the label, so are not tightly connected to anything. We have the dorsal arcs, so transversal arcs, which move backwards, as nicely seen yesterday, and that we have a ventral stress fibers reaching from the focal adhesions in the front to the focal adhesion at the rear. Then we have the keratins with the keratin forming particles, or the keratin filament precursors, where the assembly starts somewhere in the lamellipodium. We have the network, which assembles at the front, disassembles here at the end. We have the stable cage of keratin filaments around the nucleus, and we have the rear net. We do not really know what's going on here in a migrating cell.
Is this network, keratin network, distributed all over the place, all over the volume, or is it tends to the ventral?
No, it's all over the volume, and when you somehow you have the impression that you have a basal layer and a more dorsal layer.
And
if you go in 3D.
And in between still keratin. You have ventral layer, dorsal layer
of keratins,
and in between still some network of keratin.
Some network, but maybe it's concentrated to
less
the
density.
Sometimes in 3D you have the impression, yes.
How is the motility affected if you knock out keratin? Because all the models we've seen don't have that in there.
There are cell types, there are cells available which do not have any keratins. These are keratinocytes which have been, where the whole gene cluster has been knocked out, and the other half of the sibling cannot form filaments because it doesn't have a partner, so it's degraded. So these cells do not have any keratins. These cells are viable. The animals are not viable because they have defects in the embryology, in the early phase, because to transport defects, but the cells are viable. The cells even move. They even, perhaps, there's some indication that they even move faster than the control cells, which might be that the whole structure of the cell is somehow weakened and the cell is able to move faster, at least in a petri dish. No idea how it will be in a confined environment or something like that. Things are still puzzling here. But the keratin filaments are not necessary for the whole migration story. That's right. They are modulators of the. So somehow the actin filaments are pushing the cell forward. And our idea is that the keratin network coming behind is stabilizing the whole cell. So migration is not only pushing forward, but you have to get the whole bulk of cell behind it, and we think that their keratins are important for it.
So do cells leave behind more blebs when they migrate with no keratin?
Nobody knows.
You don't see fragments of cell left as the cell migrates.
Nobody knows. It's the answer. You should look at it, yes.
Nice.
So this is when when all comes together. Anyway, I'm not expecting that someone will model everything here. But perhaps we will end up with modeling some aspects of this whole story. Yeah, finally, nevertheless, all things have to team up just for migration. This is not only true for the cell, but only for researchers. So also we have to team up with others, with Rudolf Merkel, Bernd Hoffmann, with Thomas Manni's group in Leipzig, with the Lehrstuhl of Bildverarbeitung or the image analysis group at the RWTH Aachen University with the EVEs1, which made the magnetic tweezer, especially with Schlagenberg and Chen Su, and in our institute, Rudolf Leubel, Martin Mock, who made this speed analysis, and Ursula Wilhelm, who is keeping the cells alive, happy and glowing. Thank you.
Time for open